three technology, and among the three, one technology has been taken by Reliance. It is approved for the grant. 14 research paper I am having, 3 patent, and I am having several membership like Fellow of Institute of Engineers, American Institute of Chemical Engineers. Now, this your this presentation, that is some lipid-based biofuel production from wastewater. I'm presenting. I'm sharing my knowledge domain. Though this field is niche to me, I have not done much work, but some paperwork is still there. So this is wastewater, maybe from the industrial wastewater, maybe the domestic wastewater, or agricultural wastewater, as the case may be. It contains several microorganisms, and every microorganism having their own range of specificity of substrate, and also the nature of the, and also the means different types of products they produce. Now the question is this product preparation by the by the microorganism of the wastewater treatment pond. It is happening time immemorial, but recently for for water treatment also, and for waste valorization also, this scientist has taken very much interest in different type of activities, mainly for the valorization or value-added product preparation, at the same time for contribution to the green environment. Now, this wastewater, what is the advantage of this wastewater and the microorganisms living inside that? This is, first of all, wastewater treatment we can do better way by utilizing the contribution of the same microorganism who are living in that pond, and some of the nutrition and some of the nutrients and contamination nutrients from the unconsumed food material or unconsumed other materials, even from the food industry or some nutraceutical industries, and at the same time some contaminating agent, just like the pseudomonas or something like that, that is to be removed, and the nutrients can be removed and recovery. And this, as 40% of this wastewater, it has been observed by by different scientists. That is, most of the this type of waste contains 40% lipid. This that is the highest presentation of some component, that is a lipid. This higher biomass and lipid productivity, and this reduced greenhouse gas emission by because of the industrial waste, some of the carbon dioxide or carbon dioxide related derivatives are there, and these are the feedstock for the biodiesel production, because this uh, fossil fuel is coming to an end. There is the urgent need for biodiesel production. So among the biodiesel, this biodiesel, then at the same time, whenever biodiesel is produced, some side by side, some that is mainly bioethanol, biohydrogen, and biodiesel. And from the biomass, it is the nutrient and recovery and bioplastic. And finally, you will be getting the clean water. This water scarcity is slowly become another problem in the globe. Now, this biofuel, what we may produce, what we may manufacture, what we may convert the this waste to biofuels, it is divided basically in 3 generations: first generation biofuels, second generation biofuels, and third generation biofuels. First generation biofuels, that is, first generation biofuels will be coming from some of the example as the biodiesel, corn ethanol, and feedstock is particularly vegetative oil. But the industry where the vegetative oil production is going on are corn, wheat, rice, potato, rapeseed, different food preparation, food industry. from animal fats, sugar cane, etc. So these are the feedstock from this type of product we can get. But the problem is that sources of this feedstock is limited, and benefits is like that same benefit. There is environmental friendly, and it may be economic. It may be some social security because this less waste load on the earth is for better health. And there is a second generation biofuels. Second generation biofuels, mainly this feedstock is camelina, jatropha, agricultural waste such as cellulose, food waste, aquatic biomass. And there also you can get several fuel products, biofuels, bio oils, FMA, FAME, and FMA, that is fatty acid methyl ester, lignocellulosic ethanol, butanol, etc. Problem is that this process is very much lengthy, and if the process is very much lengthy, there will be the involvement of several unit operations or several utility, so it will be expensive as well. Benefits is that not directly affecting the human food chain. They are not going to destroy the human food chain, and they are not going not utilizing the fertile soils, because this jatropha is to be is to be obtained by agricultural means, and they have to get it. And next is the third generation biofuels. Third generation biofuels are coming particularly from algae, microalgae, fungi, and there also we can get the same products: biodiesel, biogas, biomethanol, hydrogen, and benefits will be this non-agricultural lands and higher oil production per unit area, because this the algae from the algae cell by extraction, human medical food extraction or something like that, we can do it. And short harvesting cycle, because their cycle is very less, we can harvest them as early as possible. Now, this is this. There are several this waste comprises 40% lipid, and that lipid is the highest representation. Now, before going to the detailed discussion, here is a general mechanism. There are particularly three, four mechanism which is happening. This lipid is there, means definitely some lipid consuming microorganism must be there, or some lipid accumulating organism must be there. This is a general mechanism for the biorefinery column for biofuel production from wastewater, and mainly they are in majority they are the anaerobic fermentation, anaerobic conditional process, and this lipid accumulation and lipid enrichment inside the organism is there. This here uh, many abbreviation will be coming in the next slides. And I'm getting some of the abbreviation here. This is FAEE, that is fatty acid methyl ester. FAME, fatty acid methyl ester. HAME, hydroxyl alkonate methyl ester. PHB, polyhydroxybutyrate. POL, PHB polymerase. TAG, that is triglycerol, and that is a wax ester. These several the abbreviation will be coming in my next slides. Now I am describing the general mechanism of the biorefinery. How does it happen? This if some substrate is there, then that substance that from that substance the lipid accumulation will be there inside the cell, and usually what happen, that lipid is there in the wastewater or municipal water or your waste, and there are several microorganisms, and on the microorganism, several extracellular lipases there. It has been observed around 7 to 8 different types of lipases are there. Why several types are there? Because this lipid, that is the basically a fatty acid agglomeration, these are from C4 to C20, different molecular weight, different structure are there. And from that fatty acid, several varieties of lipids usually formed, and this organism keeps the higher range of specificity so that any lipid comprises of any types of fatty acid accumulation, maybe C4 or C20 or whatever it may be in this range, they can consume anything. So there are several types of lipase are there. They are the extracellular lipase. They are on the upper part of the cell, that is on the extracellular, and they are responsible for this breakdown of this to the monomeric units. And then again, there are several synthase enzymes which will which will help in accumulating this lipid inside the cell. Now, one is a tag accumulator. This uh, this is triglycerol acyl. Some synthesis there, some enzyme is there, and they will be particularly using. They will find application in WS, which is the particularly wax ester or diglycerol. This is enzyme is triglycerol acyl enzyme is there. That is also transferase. This T is for transferase, and they will be responsible for this lipid micro droplets preparation, and this will conglomerate, and they will be inside the cell of the microorganism, and when it, as per the requirement, it will be accumulated, or sometimes it may be released. This is one process. This is one process where they will work. So these all this happens with the in the kingdom of microorganism in the wastewater. So in any influent wastewater, finally after all these things happenings, you will get FAME and FAEE. FAME is fatty acid methyl ester, and FAAE is fatty acid ethyl ester and fatty acid methyl ester. They will get all these things. Same thing, this it either DGAT or some enzymes are there for the wax ester. Again, this accumulation will be there. There will be the docking, the same mechanism with broad specificity of the substrate. This lipid micro droplets will be formed and it will be accumulated. Now, these are very much, in many cases, they are very much general, but there is some particular, this thing, particular polymer is there, polyhydroxybutyrate, that is there in the family of polyhydroxyalconate, PHA. In the PHA family, PHB is the most suitable, and PHB, after formation, it is having many advantages. It is one of the cheapest biopolymer available now. This it is selling price is around $2. This thing, and very easy to manufacture through fermentation. Other than that, it is also available for the biofuel preparation. This biofuel manufacturing. So PSB is most suitable candidate. It is having several application, biopolymer application, and at the same time of the this biofuel and preparation of several kinds. So in the municipal waste, if I get some more detailed discussion in this, we can see that is this municipal waste is particularly 40% lipid content is there as the total organic fraction, and most of the lipids are in triacyl glycerols format, and a minor part is the free long chain fatty acid. Some of them are long chain fatty acid also, either long chain fatty acid or triacyl glycerols. This for this biodiesel preparation or biofuel preparation, the direct synthesis from lipid-rich biomass happens. Some special customized organisms are there which occur within the biological wastewater treatment plants and accumulates copious amounts of intracellular lipids. How they are forming in my previous slide, how this biorefinery acts, I gave some bit idea. Now these are called the uh, oleaginous microorganisms. They assimilate of the lipid from wastewater or synthesize them de novo from other carbon sources and store them intracellular in the form of TAGs, OX esters, and polyhydroxy alkanoids (PHN). This uh, glycerol, basically, this is the glycerol main backbone, and there the fatty acid chains it happens. So it becomes the glycerol acyl or diacyl structure, as the case may be, and this organism now. This organism, when they have identified, in many cases, again they are bioengineered, and they produce some specific biocatalyst prototype organism or the cell which will produce the wax esters, and wax esters particularly is in the application of cosmetics and biolubricants and other applications also from this. Or there is the acetyl-CoA, that is the fatty acid synthesis from acetyl-CoA, and this is some part of the heterologous pathway engineering, how they work and how they go up to this acetyl-CoA, then finally they go to the wax ester. How do they go and how do they form? This is some of the idea. The main thing is that up to this slide, it is like that this waste contains majority in lipid, and this lipid either they are from there, some of them are existing there, and there are several organisms customized for this purpose. They either again regenerate some lipids or this fatty acid components, and they assimilate and accumulate inside the cell. And best, so far, the best applicable material is a PHB. Now, there is one process which needs some mention here. That is the chemical transesterification that also happened here. This scheme shows what does what how does it happens. So and what can be manufactured, what we can get. We can get bio oils. We can get this other biofuel, biodiesel material, and some fatty acid ester or waste ester. This triglyceride or methanol, what may be the case, by transesterification, this chemical process which has been developed basically with the main chemical reaction of just 1 second. Uh, yeah, it will come in some of the next slide. Um, it is the modification of a named chemical reaction. It can be there in the this next some slides. And this transesterification is the chemical means. And by transesterification means usually This FMA, the approximate yield of 10% FAME, fatty acid methyl esters per day per dry weight of sludge, and from it, whatever biofuel can be produced, which is coming out approximately 0.2. This is some mistake. $0.2 per liter. This is coming as the cost. This biodiesel formation. The availability of this material may be less, but however, the process is cheaper, and the material is the what is the this deliverable from this. This is also very much cheaper. Now, with this, if the this IC engine of the motor can also have some modification, we can get a very cheaper transportation system, very cheaper means materials or tools for various purposes. And if this is taken properly and this is taken care properly and proper scientific outlook is there and some wise decision may be there, then the estimated that this integration of this lipid conversion by transesterification process, 50% of all existing municipal biological wastewater treatment plants in the United States, which will produce equivalent to 0.5% of the US yearly petroleum diesel demand. This figure again can be improved by this projected improvement in the IC engine efficiency, and then this wastewater or municipal water can be a this thing boon for the society. And this process in US, people are already working on that, and some successes also there. So this is the actually origin of our discussion today, or this rationale of our discussion today. Okay, this wastewater treatment plant can be a good source of the this biodiesel and other fuel demand, and it can meet up this some agreeable and noticeable part of the consumption. This microbial lipid assimilation, and there is also another discussion with some of the this biochemical pathway. Now, these organisms, lipid accumulating organisms in BWWT plants, they have identified as the candidatus microthrix parvicella, or in short, it is called M. parvicella or microthrix parvicella. Another one is responsible particularly for PHB formation, PHB accumulating organism. It is this is called the A. Phosphates, candidatus accumulibacter phosphates. This lipidic wastewater derived biofuels such as what we get, that is bio oil we get, and several other valuable hydrocarbons, and with practical application it is there. Yeah, this is the process, this Fischer-Tropsch process. It is with some modification it has become this chemical, the main principle of the basic principle behind this chemical transesterification. So Fischer-Tropsch process, they have modified and they have done it. Now this, if you see the biochemical pathway of these two organisms, and if we properly see, then we can really understand that some gene expression is required for a dynamic EVPR condition. So this two biochemical pathway has been discussed here. One is for the anaerobic biochemical pathway, another is the aerobic biochemical pathway, and these both are showing how it happened. There is M. parvicella. This is also from M. parvicella, and how this mechanism happens, and finally we are getting the frog formers and what going on inside this, and this is for the aerobic way, and we shown this this small pathway is showing how PHA and PHB forms from this for PHA formation. These two genes are responsible, PHA and PHC, and from that we can get the acetyl CoA, and again for citrate, and finally we are getting this glycogen also. So for PHA formation, this glycogen also there, and here there is the PHA formation. From this is the basic backbone of the biochemical pathway, and these are the two products. This is another this thing, another some pathway, and where we can see how this tag and work sister forms, and what happens. It goes through the TCA cycle also, and the basic fundamental platform material is the acyl CoA. Anything to form acyl CoA first, and from that acyl CoA different other products, and from the different other products you can get the desired product also. So whatever fatty acid, the agglomeration of fatty acid, finally lipid you are getting, it will go first for acyl CoA pathway, and from acyl CoA there is some fatty aldehyde, and there also you can get the this wax ester, or there is another thing to acyl G3P, and this is the diacyl glycerol finally, and through that you're getting the triacyl glycerol. There is another, and some part also responsible for the, so some part is for the TCA cycle also. It will go to the side of the acyl or acetyl CoA here, and there is some other cycle EMP, and how this glucose 6P you are getting acetyl CoA, and then again it goes into the TCA cycle. So acyl CoA, acetyl CoA, these are basically the basic platform of the material where this lipid consumption for the lipid this thing from a lipid accumulation or lipid synthesis, or whatever the products required. So this is some of the cycle also having some explanation how it happens. That is from the lipid, what are the products forms and how it forms. See, this is now it is the polyhydroxybutyrate formation, and polyhydroxybutyrate is the most important product here. It will go for the biopolymer as well as some of the materials for the biofuel production. This pH or the particular customized PHB, this bacterial lipid inclusion inside the cell happens, and this lipid inclusions are called carbonogens also. Carbonogen consisting of PHA. PHA they are basically for the replacement of the petroleum-based plastic, the bioplastic. Now there are other applications the alternative use of pH, which includes the production of this is hydroxy alkanoids, methyl esters, which can serve as the biofuels or as fuel additives. Then from pH in the pH family, PHB is most known. Polyhydroxybutyrate is typically is the most abundant pH among pH. PHB is the most abundant, and many studies have been done here from decades. Uh, that is the reason today PHB is available in the market by just $2 per kilo, and the genetic modification or genetic studies of the this PHB formation and what is going on for the enzymatic reaction. It has been studied very well. The gene encoding is the key enzyme involved in the synthesis of monomeric precursor of PHB, and then there is the polymerization and depolymerization process. So this monomeric precursor forms, and further some enzymatic reactions or enzymatic transformation is responsible, and that is the reason we are getting the PHB. Now, our current understanding based on work is this is some of the PHB also works in the scaffold model. This current understanding based on work on model organisms as the in the scaffold model in which PHB is synthesis attached first and then scaffold molecule to produce initiation complex or PHA biosynthesis. These scaffold molecules are mainly DNA or some PHA granule associated protein PHAM. This also aids in the dimerization of pH synthesis, and protein other proteins coating pH granules have also been identified in that. And in this process, this is actually polyhydroxybutyrate. There are many big industries in US nowadays, and this several tons per day is the production capacity. But yield comes around 50% of the. It is this. Uh, this is quantified like how much percentage of pH available in the total shell dry weight after fermentation. This shell dry weight is taken. This broth fermentation broth after filtration, this liquid will go, and from in the shell dry weight there is some other material along with PHB, and how much percentage of the dry weight contains PHB, that is the measure, that is the quantitative measure in this case. And in this process, this anaerobic process, and if from this BWWT bacterial community, and this process, if it is adapted, it will give 77% of the dry weight. Although present the present production rate is around 50 to 55% of the pH. So this much only. This is this lipid based biofuel production. How lipid based biofuel, what is the process, accumulation, and then aggregation, and then formation, and some of the biochemical pathway involved in that, and how do they work, and how the genetic coding or genetic engineering work activities have been done. So um, uh, this is the studies so far. The summary of this summary of the studies so far. Now I am in CIAB, and with me these are my students are presently working with me. So this is all from myself.
Thank you, sir. Thanks for your presentation. You explained nicely here. We will whatever you worked in the CIA. since I think last 4 or 5 years. How many years? I hope it is almost 4 years or so, right, sir?
Yeah.
4 to 5 years you have it. Now it is open for the discussion. So, participant, if you have any doubt regarding this, you can easily ask Professor Dr. Goswami. He will obviously explain it. So, anyone have any doubt? I don't think anyone have any doubt. It's already self-explanatory things are there. Sir, it's a kind request from the participant that you kindly share your video with us so that later also if they find they will mail it.
Hello?
Can you hear me, sir?
Yeah.
Yeah, actually, participant want your this video means this presentation with them. They will just learn it, and if they find some drawbacks or find some information, more information, they will also mail you. So, and I
Okay.
will also.
Yeah, you can share it. And this is basically some of the review type preparation
Yeah.
and I have collected from many papers and all, and this is the accumulation of that. So they can use it for their own purposes. And in maximum cases, I have mentioned where from which review paper I have collected that. It is also there. But this some of the materials, it is not my basic research work. It is basically the accumulation of the research paper from others. Okay.
That I understand. Uh, Participant, kindly share that video with me so that I can also share with them. So this is all. So anyone have any question, kindly ask. Okay, thank you, sir. Then thanks for giving us time.
Thank
Sir,
you, Dr. Sasmal, for
sir, one
providing
question
me.
is there.
Yeah, yeah,
One
yeah.
question is there. Some one participant asked, what is the CFU for optimum production? Dr. Jyoti, can
This
you?
can you ask the candidate
Yeah, yeah, yeah.
for
Dr.
video? This can the candidate on the
Yeah,
camera
yeah,
and video so that I can see face to face.
sure, sure. Dr. Jyoti, can you just unmute your video and mic so that sir can answer your question and
Good afternoon, sir.
yeah, yeah.
Good afternoon, ma'am.
Yeah, so I would like to know the CFPO, for optimum production of biodiesel.
PHB,
For
optimum
optimum
production of
production
PHB,
of biodiesel, sir.
optimum production of biodiesel. Biodiesel is very much general term. See, the fact is that for some particular lipid and for some particular organism, there is a particular product which is called the bio-oils, and that bio-oils will be the component of the biodiesel.
Yes, sir.
So the proper studies and uh, means for several years it has been done basically on polyhydroxy butyrate
Okay, sir.
by organism by the this organism by fermentation, means aerobic or anaerobic, whatever may be the case. Or other thing, that is this Reliance algae project, whatever I am also a part.
Yes,
They are
sir.
what they are doing. They are simply this algae they are growing on the pond, and
Okay.
they are taking the extraction of the biomass, and they are getting some oil. In that case, this recovery comes around 25 to 30%. This is oil from that biomass, and in that the purity is 40 to 45%.
Sir, is there any specific kind of algae they are harvesting or it is mixed consortium of algae?
Yeah, they are having this Reliance. I can't discuss much in details
Yeah.
because I'm
Yes.
in agreement.
Yes.
The fact
Yes.
is that they are in Jamnagar. They are having the ponds, and from the ponds, several types of algae they do culture and they do harvest. And they send me some dried algae leaves, but they did not disclose the name. And from that, my observation is that around 25 to 30% oil can be extracted. Basically, they are all lipids.
Yes, sir.
And these lipids are they are not very complicated lipids. The algae selection they have done so wisely that whatever maybe the lipid will be extracted from that biomass, it is not very much complicated because they again don't want to do the hydrogenation.
Yes, sir.
All the biodiesel or all the petroleum, whatever we are doing, we are getting it. There is one step is the hydrogenation. So people don't want to do the hydrogenation. So simple lipid they are getting. That algae they choose very wisely. So you can take on average, if it is pure form, it will be 8 to 10%, and if it is in the crude form, it will be around 20%.
This is a yield.
It is the
Thank you.
yield. Yeah.
Thank you, sir.
Okay.
What about the quality of oil you are getting? Means its splash point and other things whether you are checking, sir?
Basically, so far there are many studies going on, but what is practically feasible now that is the DHA, that is decahexaenoic acid, and decahexaenoic acid is having very good application. Even
Development of brain.
this can act some of the biological replacement also. That is the your liquid inside your eyeball or some other this like the gelling material in your orthopedic structure. So that they are using, and that is basically DHA, and mainly the yield is the pure yield is around 8 to 10%. But if you take the crude is also having several applications. Crude is around just 25 to 25%.
Okay, so it is almost equivalent to your oil production.
They have to go for high production, but thing is that with this type of this around 10 to 15% yield, it is also keeping huge profit margin.
Okay.
Yeah.
This is a new aspect we learned that okay from the
Because the cultivation of algae basically needs some particular wavelength of the this thing, whatever light available. So and sunlight is very much matching with that, and the salinity of the pond. Salinity of the pond, it is at our hand only. No, we can maintain the salinity, and the main thing is the light. Uh, mm, this light, whatever sunlight is available, that is matching with that. Or artificially, if the operation is to run 24 hours in the night, there is some photobioreactor. They are maintaining some particular frequency and wavelength of the light, whatever they do need. Even though it is a very profitable process after all these things.
So is there any such kind of photobioreactor is present in CIAB? So that all your faculty they can send their the students also means what kind of facilities available there for further training that kind of thing.
This photobiotic reactor, Reliance is having, and then NTPC is having, and even this Hindustan Petroleum